So thanks for asking me to speak, but Suzanne, I have a bone to pick with you actually, because, well, first you're late, so. Second, my tag says Boston University, and there's trade rumors swirling. I don't know what's happening, but okay. Yeah, so something's happening. So I better do a good job, or else, what happens? Okay. Yeah, well, so it goes. I'm tired of the sunshine anyways. Okay. Yeah. All right, can we be serious? Let's be serious. So, all right. So, the life and times of RNA. You've seen a lot of descriptions today about the type of functions that RNA can perform. And some talk has been given to the fact that secondary structure and often tertiary structure elements are critically important for regulating the biological function of RNA. So for example, long non-coding RNAs, which Mitch is going to talk about in a few minutes, can interact with proteins and regulate chromatin. Long repeat elements can form long double-stranded regions in many neurodegenerative diseases, and their role is to sequester proteins or splicing proteins in the nucleus. But outside of the nucleus in the cytoplasm, there are a bunch of structure elements in RNA that control its ability to interact not only with itself, but also in transactive factors like proteins, small RNA protein complexes like Ian was talking about earlier today. And structure elements in untranslated regions are very important for regulating RNA biology. So when it comes to RNA structure in the cell, there are a lot of questions that remain to be addressed. For example, does the structure of an RNA inside of the cell and outside of the cell look the same? Are the thermodynamic rules preserved? Or is it changed and manipulated by other elements inside the cell? What are the general principles of how RNA structure controls RNA biology? These are big questions that remain in the field. And if I can make a corollary to work that's been done on chromatin, we've learned a lot about chromatin and the way genes are regulated by studying the structure of chromatin. And that's been done by using DNA split printing, for example, or small molecules formaldehyde cross-linking is another example. But we've learned so much about the way the three-dimensional organization of the genome is encoded, and secondary structural organization is encoded by doing that. Now, that has its roots in biochemistry, so studying one stretch of DNA at a time. And the RNA field has a lot of its biochemical roots in doing very similar experiments, studying one RNA at a time through very controlled and exquisitely elegant, but argue, biochemical experiments, where you're studying one structure at a time in RNA. And if we think about genomics as the same example, using genomics to understand the genome, we now understand some of the guiding principles that regulate thousands of genes inside of the cell. So we asked a question when I was a postdoc in Howard Cheng's lab, can we do the same thing with RNA biology? We started to extract some of those general principles by studying the structure of thousands of RNAs at a time. So back to the biochemistry, okay? So here's where the heart is. In structure probing experiments, there's really two flavors. So you can study it using RNases, which are protein enzymes. And those RNases are very good at recognizing structural motifs like single-stranded stretches or double-stranded stretches, but they lack in the ability to perform probing at single nucleotide resolution because of three-dimensional sterics. Okay? Another class are small molecules or chemicals, okay, in which chemical reactions occur between reagents and the nucleobases or the backbone of RNA itself. For example, dimethyl sulfate is the classic alkylating reagent that will react with the N1 position of adenosine, N3 of cytosine, or N7 of G in some instances. And the information that you get from that is if I have a chemical reaction that occurs with the Watson-Crick base of adenosine, that Watson-Crick base is not base paired. Okay? Because when it is base paired, the chemical reactivity is sequestered by sterics through the Watson-Crick interaction, but also lack of chemical reactivity at N1. Right? So chemicals can react with RNA, form covalent bonds, and leave a print on that RNA. And the next question is, how do we identify that print? In normal experiments, you would fold your RNA, modify it, perform reverse transcription, and reverse transcriptase will stop creating a cDNA molecule that's complementary to the RNA sequence that it was primed against. All right? So you get two pieces of information that will become very important in the experiments I'm going to talk about in a minute. The first is you get a length. Okay? By normal gel electrophoresis, this is how you infer where the reverse transcription stop was. The second big piece of information you get is genetic. So it's cDNA, the complementary DNA sequence, which we can later use in the context of genome-wide sequencing. Okay? And you get a band of reactivity, and this reactivity is scaled to how many times my reverse transcription stopped, which is related to how many times that chemical reaction occurred. Okay? In things where there's a lot of chemical reactivity, it's very single-stranded. Things where there's little to no reactivity, it's double-stranded in most cases. All right? Now, the other thing I'll point out in this type of experiment is that reagents have high selectivity. Most of them are not generalizable. So they will react with lots of interface of certain nucleotides, or will react with different heteroatoms, and they're not specific to points that are structure elements in RNA that are preserved in every internucleotide linkage. Recently, there's been the development of reagents that can do that, and the gold standard seems to be running up to be SHAPE. So SHAPE is selective hydroxyl acylation, which you then analyze by primer extension, shown here. Okay? So in SHAPE, the 2'-hydroxyl is a chemical modality that's preserved in every internucleotide linkage. And there's something special about that that you can relate to secondary structure. The problem with most SHAPE reagents is that they're not soluble enough that they work inside the cell. So their low solubility becomes a problem because there's a limited amount of reagent that can also be souped up by other nucleophiles that are present in the cell. So there's a substantial amount of competition between my RNA and my other nucleophiles, especially in the context of nucleotides, which can have their concentration be in the millimolar range, 10 tens of millimolar, depending on the cell type. So a big problem. When we were thinking about this in terms of chemistry, we, through screening many different molecules, came up with this class of nicotinic acid acylimidazole reagents, which I'm showing here. Okay? And SHAPE works by sensing the chemical environment of the 2'-hydroxyl. In RNA, if the nucleobase is single-stranded, the internucleotide linkage has a high degree of entropic energy. It's flexible, sampling many different conformations. And on a rare instance, the 2'-hydroxyl will come in close proximity to the phosphate, and the proton will start to be transiently deprotonated, activating it as a nucleophile. And that nucleophile can react with acylating electrophiles to form this covalent print that I mentioned today. Okay, so just as a reminder, these hydroxyls that are being acylated are in single-stranded regions of the RNA. So when we first showed these class of chemicals, we started, just like everybody else, in trying to understand their chemical reactivity in very controlled systems. And our favorite RNA of choice at the time was 5S ribosomal RNA, because we could study it inside and outside of the cell, try to understand how a reagent was understanding the structural intricacies of the cellular environment, and try to extract what those chemical reactivity profiles were telling us about the biological function of such an RNA. So here's an example of a reverse transcription DEL with a radiolabeled primer. So here's my ladder. Okay. Here's my DMSO negative control. Here's my reagent if I reacted in vitro. And here's my reagent if I reacted in vivo. So you can see just by a glance at the reactivity pattern, every time that there's a band, that RNA was modified at that position. Okay? So therefore, we infer that that's single-stranded. When we compare not only the secondary structure of 5S ribosomal RNA to our reactivity program, or profile, but also the three-dimensional structure, what we find is that our reagent is measuring secondary structure exactly the way it's supposed to. Not only that, but these new acylating reagents measure secondary structure in exactly the same way that the first group of RNA acylating reagents developed by Kevin Weeks' lab do. Okay. So good, a couple of controls working out. When we think about the biology, the residues that are different, so their structure profile is different, if we map them onto the crystal structure with 5S ribosomal RNA, as well as some EM structures, what we find is that points of difference, okay, things that are different inside and outside of the cell, map to points on the RNA structure where the RNA is interacting with other molecules, such as other ribosomal RNA, or in this case, ribosomal protein L5. Okay, so that's one thing. So we're able to actually understand how the 2' hydroxyl environment is changing inside and outside of the cell and how that can be contributing to chemical reactivity. So this tells us our reagent's measuring the bona fide RNA structure inside the cell. The second thing was, if we compare all these elements that are changing, all these nucleotides that are changing their reactivity, and we look in the literature of saturated mutagenesis and conserved residues in yeast, what we actually found, which I found to be very surprising, was that everywhere that there was a difference, those residues were found to be important for regulating translation rate, translation fidelity, and the maturation of the ribosome inside the cell. Which means that this structural state that we're measuring with these acylated reagents is critical for how that RNA is going to behave. So very important. So try to work through these controls and make sure that we're doing the right thing. Okay, so these reagents I showed you before. Okay, right here, this methyl in the 2 position next to the nitrogen and the nicotinic acid. I want to tell you the dirty secret. The dirty secret is, if you look at this gel, there's a lot of reverse transcription stops, right? But it turns out the one that's full length dominates the signal. About 70% of the signal comes from RNA that's actually not modified. Now imagine if I walked into your office in UCI and I was excited about this, what would you say? You'd never do a genomic experiment on that because the majority of your reads would be junk, right? Because if we want to map reverse transcription stops, a lot of our signals not only going to come from spurious RT stops, which reverse transcriptase is prone to, but also a lot of that signal is going to come from unmodified RNA, which means that the sequencing complexity is going to be dominated by false positives. Okay? So, how do we get over that problem? To take this into a transcriptome-wide measurement protocol, we thought one potential way to do that would be to develop it in such a way that we could isolate all the points of acylation. So now our reverse transcription profile would be dominated by RT stops that are due to chemical probing, right? So we developed this bifunctional reagent shown here. So the nicotinic acid acylimidazoles is preserved, that structure, but there's also an azide at the 2 position, appended at the methylene position, okay? Now, the reason that's useful is because we can use cyclooctines to do copper-free click chemistry and pull out those RNAs that are modified with a biopsy handle, okay? And we worked out all the controls for this protocol where you can do RNA isolation inside living cells. You isolate the RNA, perform this click reaction, you isolate your RNA, and you can do reverse transcription on a resin. So the purity is very high because you can wash away all those reverse transcription stops off of the resin. And because you're using a bioacylated handle, shown here, our washing conditions are 5 molar salt between, you can just beat it up pretty good. You get rid of everything. And we've done a ton of controls to show that if you do this experiment with a radiolabeled primer, there's literally no signal for the samples that don't have this biopsy handle on them, which means that we're getting rid of all the stops that are not due to chemical modification. So signal-to-noise is a big issue, especially in transcriptome-wide data sets. The first thing we did was compare our RNA structure stops to known crystal structures, again, relying on the ribosome. And we're showing that our chemical modification profile was essentially the same as the profile you get from monitoring this 1 RNA at a time in a gel in a lab setting. Okay? So we can recapitulate the secondary structure profile by shape of the ribosome in literally 3 days, where it would take months to do that. We can compare our shape sequencing data to DMS probing as well, and we showed that we get really strong sensitivity with shape when you compare it to the B factor of the crystal structure. So that's the Debye-Wally factor, which is telling you the thermal energy or thermal motion. Okay, so that's sort of a proxy for flexibility inside the crystal. That makes sense because our reagents are measuring that high entropic value. Okay, so they should be highly correlated, which we observed in the crystal structure shown here. We next were able to show that we can recapitulate known RNA secondary structure elements in RNA in messenger RNA. So these are the more lowly expressed RNAs inside the cell. So if you think about it, 95, 98% of the RNA content inside the cell is ribosomes. We're actually sampling structures of lower expressed messenger RNAs with high accuracy. Here's just a couple of examples in comparing, those structures inside and outside of the cell. We then wanted to take a closer look at this. So we wanted to ask the question, if I have a structure, I'll make one point on this slide first, is that inside the cell, we have been able to demonstrate that these reagents can measure RNA structure in the nucleus, on mitochondrial RNAs, in the cytoplasm, etc. So it seems like reagents are just being able to disperse everywhere inside the cell and give us a pretty strong structure profile. So here's an RNA that's associated inside the nucleus malum 1, a long non-coding RNA. In vitro, we get this RNA structure profile, and in vivo, it seems to have a much higher level of shape reactivity. We then wanted to know, if I do have structure differences, where do those differences arise? And it turns out that adenosine residues have quite a difference in their structure flexibility inside and outside of the cell, whereas G's and C's seem to have overall preserved of their structural flexibility, which may not be surprising because G's and C's are more apt to be in base paired regions in RNA. Okay? So this BTD that I'm showing here is the difference between in vitro and in vivo. Okay, so because we were able to ask that question, what things are different, we wanted to know, is there any biological association with elements that would be different? So we asked that question in the context of hexamers. And what we found is that things that are associated with post-transcriptional regulation seem to have large differences in their shape reactivity profile inside and outside of the cell, which suggested to us very early that this difference could actually be due to protein binding and structural rearrangement. Whereas in comparison, the Kozak sequence near translation start sites seems to be preserved, the secondary structure seems to be preserved inside and outside of the cell. So our hypothesis was that perhaps we can exploit these structural differences to actually predict things that are interacting with RNA, to predict changes that are happening on the RNA structure due to transacting factors or even chemical modification. All right, so our test case 1, translation start sites. We looked at the Kozak sequence, the VTD was very low. Okay? So here's our start site in vivo and start site in vitro. AUG start codon seem to be very similar in their structure, which is consistent with our genome-wide analysis that I showed on the previous page. And we also were able to do this for U-orbs and N-terminal truncations in vivo. So all this data comes from, or these coordinates for translation sites come from ribosome profiling data that was generated by Nick and Golia in the same cells. Okay. So another test case is ribosome pause sites. It's been hypothesized in the literature for quite some time that secondary structure elements can prevent the ribosome from having efficient read-through, which is surprising because of the intrinsic helicase activity of ribosomes. So when we look at this, here's the secondary structure profile of genes and aggregates at all pause sites. So the APE site is shown here. And there is some triplet nucleotide pattern that we don't quite understand yet. We have a few hypotheses about this, but we don't know exactly what this means. And if you compare that actually to the same primary sequences that do not have pause sites identified by ribosome profiling, you see that there is this difference at the A site where the A site is quite reactive, but that this nucleotide repeating pattern is lost, and the delta here is much different than the delta here, which is much lower at ribosome pause sites. Okay. This suggests that this structure signature may actually be important for regulating pause site selection, and also that the secondary structure in aggregate is very similar inside and outside of the cell, which suggests that the primary sequence is what's controlling this, not necessarily proteins or other factors. So, something else, of course, RNAs are rarely without a protein partner inside the cell, so we want to understand if we can actually predict or study RNA-protein interactions on the same scale. So for example, here's the RB-FOX, which is an important protein not only in RNA biology in general, but also in stem cells, the system we were studying. And here's an NMR structure showing the primary sequence threaded through on the structure, and I've highlighted the 2' hydroxyls in the structure, and that will become important in a second. So, To understand this, we generated two pieces of data. The first data was our RNA structure, okay, at those motifs. And our second was we performed CLIP on RB-FOX proteins inside these cells to identify the true positive sites of where those proteins were occupied on those RNAs. When we look at the secondary structure, or shape reactivity, difference, what we find is that the shape reactivity profile actually will recapitulate the flexibility that's seen in the NMR with different conformations that are predicted in the NMR structure. Not only that, but if we take the difference between those binding site motifs that are bound and those binding site motifs that are unbound, so true positive, false positives, with the receiver operator, we can predict greater than 75% of those protein binding sites transcriptome-wide, just based on their structure signature that we observed. The second thing we wanted to do, and this relates very closely to what Crystal was talking about before, is we want to look at things like post-transcriptional modification, in this case N6 methylation, which has recently been shown and very important to regulate not only cell biology, but the actual half-life of RNA. Okay? So again, two pieces of data, our secondary structure profiling, and also we identified the M6A sites by M6A, IP, and sequencing. Very similar to what Crystal was talking about earlier today. What we found was something very interesting. So take a look at this. When the metal-3 enzyme, in this case, is present in the cell, there's a shape reactivity profile that suggests the adenosine in what ends up being a G at the -1 position is very flexible. Okay, so when we ask the question, is this because the enzyme's there? Is this because that site's been modified and now the structure is different? We can't really distinguish between that. So we wanted to know because the field was sort of conflicted on understanding exactly how the N6 methyltransferase will recognize the RNA and catalyze methyltransferase activity by recognizing the structure of the RNA itself. So in the context of the knockout, so we generated the CRISPR knockout, like Crystal was saying before, okay? What we find is that that shape reactivity goes away, which is very suggestive that this structure element is actually base paired. Okay? So remember, if I'm highly shape reactive, I'm single-stranded. If I have low shape reactivity, I'm double-stranded. This suggested to us at the time, and we were doing biochemistry to corroborate this, that these were double-stranded regions. We found through some more targeted structure probing that this is consistent, doing some nice chemical probing one gene at a time, but it was further corroborated by another group, Chuan He's lab, which showed that adenosine residues that are methylated are found in long double-stranded regions in RNA. So our chemical probing is being corroborated by others that are doing more targeted approaches on secondary structure analysis. Okay, now we wanted to again ask the question, can we predict these methylation sites just based on the structure signature that I showed you before? And we're able to show that we can predict about 92% of them on the entire transcriptome, which is significant because, as people have said, doing these methyl sequencing experiments, it's hard to actually figure out which adenosine is modified and where the summit of that peak is. This suggests that there is another way that we can potentially use this data to predict those methylation sites as well. And I want to bring this up because it's unclear, because the lack of crystallographic data or high-resolution data, exactly how these RNAs are recognized by their proteins. And I think this is almost reminiscent of DNA modification proteins or DNA repair enzymes that rely on large sequences of double-stranded residues, and they just flip out a single base to perform their chemical reaction, which is consistent with what we're seeing for our chemical probing and what Chuan He's lab saw as well. So, some of my conclusions. We're trying to develop this new chemical probing method to predict post-transcriptional interactions and measure secondary structure in cells, whole transcriptomes at a time. And we're now interested in extending this to different cell types, different conditions, to try to understand how structure is regulating cell biology. So I want to thank Howard, who was an amazing advisor. Just let me, do what I want. And HHMI and APG Foundation for funding the NIH. And these guys right here, which were very instrumental in this. And I want to thank the people that have joined my lab because, it takes a lot of guts to join a new lab. So these are my students and postdoc Yeah, especially mine, that's right. And the RNA Symposium organizers and UCI for supporting the research that we're doing now. So.
One question.
I have a question about your translational profile.
Yeah.
So obviously, I assume you're looking at the bulk of elongated ribosomes.
Both?
The signal you get is from elongated ribosomes.
Yes.
So these
Except for
are-
the plus site. Yes.
So they will be in two states, one in the rotated and one in the unrotated.
Yes.
Is that where you see your noise or these different signals?
Um,
If so, then you can simply add, for example, cyclohexamide. That should create some channel and you should see different patterns.
Yeah. So two things. First is I love that experiment and I want to do that. Okay. The second is because I've actually suggested that because that's a great idea for sure. It is not. It is not. But it is in some of the initiation sites where we can compare that, the ribosome profiling, to that. The second thing is, I think it's going to end up being a kinetics problem actually, because the rate of ratcheting is going to be much faster than the rate of isolation. So I'm not sure how you would parse that, but I think it's worth doing. No questions on that. But I think it might end up being indistinguishable. Yeah.
Crystal, how?
So, because the N60 modification and efficient size, the percentage is very different. How sensitive is associated to disease in the N60? For example, if 1% of an RNA molecule has this modification, you detect or versus 50%?
I guess the answer to that is I don't know. But what I will say is that we are biased because the sites that we choose to look at our structure probing with the motif are those that cluster towards the higher IP signal, so which would suggest that they have a higher occupancy of M6A. So I don't know the cumulative distribution of the structure difference between lowly modified RNAs and more modified RNAs. I don't know. I don't know that. Yeah. I would suspect that it would precipitously fall off, though. Yeah, just because you're basically measuring these structures in aggregate, so you're getting an average of all of them. Yeah, the molecules of the RNA.